Well, welcome everyone. This is our opening session to our next season of our URIG our sessions where we get together and we listen to awesome science. So we're going to have a little slightly different format as we had last year, or I guess it wasn't last year, before the summer. So right now we're going to do, for this season, we'll have kind of a long talk, and then also a trainee talk for what we're trying to hope for most of our sessions. So and we're currently filling out the schedule, and we're going to be reaching out to a bunch of people. If you're interested in giving either a long talk, or if you're a trainee and you would like to give a talk, just reach out to me. I'm happy to work you into the schedule. And PIs, if you think that you have a trainee that you would like to nominate to give a trainee talk, that would be really awesome too. We would really appreciate filling in and getting some representation of our trainees. Yes, so please nominate them. And today we're going to hear from Dr. Nicole De Nisco, who is not only Associate Professor in Department of Biological Sciences at UT Dallas, but she also happens to be my mentor, so yay. And Dr. De Nisco did her PhD at MIT, and then she went on to do her postdoc at UT Southwestern. And right now, I'm a member of her lab, and we study the urobiome and all kinds of fun things with recurrent UTI. She's going to give a really interesting talk about a developing story in the lab. So go ahead. And I think if you could switch your screen to, and we'll have yeah, perfect. Awesome.
Great.
Cool.
Thank you. All right, I was waiting for someone to tell me if I needed to swap the display. So thanks for telling me that, Mike. Um, yeah, so I'm really excited to tell you all about a story we've been working on in my lab now for the past 2.5 years, believe it or not. It's been a long time. We think it's really exciting about kind of the metabolic requirements of our favorite lactobacillus species that we study as part of the urinary microbiome. So we all know that lactobacillus predominance is a hallmark of urogenital health. So a lot of groups have shown this. Michael's paper in Cell Reports Medicine, about postmenopausal women and their urogenital microbiome. We saw that women that were healthy, that had no UTI history, really had this predominance of lactobacillus species, that was further supported by estrogen hormone therapy, where we see an almost exclusive association of lactobacillus predominance in this niche with estrogen hormone therapy in women taking in postmenopausal women. So, and thinking about understanding more, on the microbiological and molecular and biochemical level of these bacteria is really important since they're associated with health. And if anyone, if you're in this field, you know that lactobacillus is actually very complicated, and you've probably seen this taxonomic note that was published back in 2020 that separated the genus Lactobacillus into a ton of new genera. And this is just a reflection of our developing understanding of these bacteria and how they differ between different species and now between different genera. And so if you've read this paper, you might remember this figure where it's showing the new proposed taxonomy for the Lactobacilliaceae family. And within this, we have distinct genera. And so we have our Lactobacillus genus. And so this includes species that you might be familiar with, like Lactobacillus crispatus. And you can see that actually over here on the tree, over here in this green clade on the left. And then we also have, we've split now from calling it Lactobacillus rhamnosus. It's now a new genus called Lactobacillus, and this includes Lactobacillus rhamnosus, KCI, paracasei, and a lot of those more generalist Lactobacillus species or Lactobacillus species as we're learning. And so there you can see that they actually are represented over here in this clade. And then we also have something that might be familiar if you work in mammalian microbiomes. There's Lactobacillus salvarius was reclassified into the genus Lactobacillus. So that's this clade over here in black. We can see the Lactobacillus salvarius is there among other members of its genus. And then I'll be talking about using as an example in this talk today. So I thought I'd point it out. There's a new Lactoplantibacillus genus that is will include things like Lactoplantibacillus plantarum and other associated species. And then finally, something that we've seen in our cohorts and is there variably, I think, in the urogenital space, Lactobacillus vaginalis is reclassified into the genus Lactobacillus over here. And so I think when this paper first came out, it was whenever there's a taxonomic change, it can be quite frustrating when you have to rename everything again in your papers. But I think in this case, especially, it actually really is warranted because we're starting to see that there are really important phenotypic differences between these different genera and even within species in the same genus. So when we're talking about Lactobacillus, right, we're not, now we have to be a little bit more specific about the exact genus that we're talking about and thinking about where it calls home, where we have, you can see over here that all of the species within the genus Lactobacillus are going to require some kind of host, whether it be vertebrate or insect, whereas Lactobacillus seem to be more nomadic species that can exist in a variety of different environments, for example. So who are the urogenital lactobacilli? And I'm sure that this is up for debate, but generally what's accepted between, I think, the vaginal microbiome fields and our urinary microbiome field is that the predominant members are going to be Lactobacillus crispatus, Lactobacillus agasseri, Lactobacillus jenseni, and the recently defined Lactobacillus mulgaris. Johnsoni, and then inners, which, you know, we might have good or bad impacts on the space. And so here's just a micrograph of Lactobacillus agasseri from my lab. We're actually developing family or genus level probes to be able to identify these spatially within samples. And so when we were starting to work with these lactobacilli, of course, we think about, okay, how do we grow them? And so the most widely used media to grow lactobacilli in general is MRS, so demand rugosa sharp media. And this is kind of what the ingredient list of what it contains. And so you see this is a completely what we call an undefined media. So it's going to contain various extracts. And so we don't know exactly what's in those extracts. So it's hard to say, to really assay their metabolic requirements when you are using these kinds of very undefined media. So we went about in the lab and we started kind of just taking things out of it as an effort to start to see what they might require. And so we wanted to kind of narrow down the components necessary for growth so we could see if we could make a more defined media for them. And so one of the first things that we found, and this is work that started a long time ago, is that tween 80 or polysorbate 80 was required for the growth of specifically the urogenital lactobacillus species. So you can see in this graph here, we have MRS, so we reconstituted MRS from the components, and we just either left out tween in the purple, or we included tween in the red dots. And you can see that all of these urogenital species of Lactobacillus cannot grow at all in MRS if you do not supplement in tween. Whereas the Lactobacillus and the Lactoplantibacilli, like rhamnosus and plantarum, really don't care if tween 80 is present or not. They grow to the same optical density, whether or not you supplement MRS with tween 80. So this suggests that they don't require this. And so we were thinking about, wow, why do all of these urogenital lactobacillus species have this absolute requirement for tween 80 in the media? And so if you look at the structure of tween 80, you can see that it's a hydrophilic polysorbate head conjugated to this tail. And what this tail actually is its oleic acid. So this fatty acid tail here is actually oleic acid. And so that's kind of hinted at what we thought might be a potential auxotrophy among these urogenital lactobacillus species, that perhaps the reason that they needed tween 80 was because they were specifically auxotrophs for fatty acids, and that the oleic acid that was contained within tween 80 was able to fulfill this auxotrophy. So we actually developed a chemically defined media, and we were able to use this to test this hypothesis that, in fact, the reason that they needed tween 80 was because of it provided oleic acid. And so we developed a completely chemically defined media, and we either added tween 80 or we added oleic acid. And we had to conjugate the oleic acid onto a vehicle, bovine serum albumin, to act as a lipid carrier protein. And so we have our CDM alone and CDM with vehicle experiments, and we show that the vehicle doesn't stimulate growth. But when we add oleic acid, and this is in the absence of tween and just oleic acid, we see that very robust growth. So we see that oleic acid basically phenocopies tween 80 in chemically defined media. And so we saw this for Lactobacillus crispatus with two different strains, LC195 and LC1903. And we also saw the same phenomenon in Lactobacillus gasseri with LG637 and LG1507. And so we're pretty convinced now, at least, that they definitely are requiring tween 80 because it's a source of oleic acid for them. But we wanted to further test our hypothesis that these other Lactobacillus and Lactoplantibacillus actually didn't have the same requirement. And so we grew Lactobacillus rhamnosus in CDM with and without tween 80. And although we see a growth stimulation in this very minimal media with tween 80, it absolutely doesn't require it. It's still required. It still gets up to high ODs without it. And then in Lactobacillus plantarum, it also doesn't care if there's oleic acid or tween 80 there. So we see the same phenomenon, that Lactobacillus rhamnosus and Lactoplantibacillus plantarum do not require oleic acid. So that led us to our hypothesis that urogenital lactobacillus species cannot perform de novo fatty acid biosynthesis. So this is the canonical FAS2 biosynthetic pathway that's encoded by the Fab operon in many gram-positive bacteria, including Staphylococcus aureus, and where it's been more elucidated, and of course in gram-negative bacteria too. It's pretty well conserved. And so we can see all the enzymes required for basically synthesizing fatty acids from acetyl-CoA into eventually becoming phospholipids. And so we decided to first use bioinformatics to take a look to see if these enzymes, or if the Fab operon that encodes these enzymes was actually conserved within the genomes of of our urogenital lactobacillus species. And so what we found using gene synteny analysis, and so if you're not familiar with gene synteny, it's basically looking at the preservation of the order of genes on a chromosome from a common ancestor. So we're looking at the conservation of the order of the genes of the Fab operon and its actual genetic location. And so this is a bit more precise than doing a gene presence, gene absence presence analysis, because that can pick up very closely related genes, especially if there's domains that are very common in proteins of different function. And this gives you more of an idea if that actual, in this case, biosynthetic unit is really conserved within a species. And so we did this gene synteny analysis for many members of the Lactobacillus genus, Lactoplantibacillus genus, and Lactobacillus genus, as well as Lactobacillus vaginalis. And we were able to see that all of our kind of non-urogenital lactobacilli here were what we call synthetic for the Fab operon. So that they contained the Fab operon, and it was in synteny with common ancestors. Whereas all of these lactobacillus species here, actually including Acidophilus, actually we're lacking synteny. So we have the Fab operon is present in what we call non-lactobacillus lactobacilli, and urogenital lactobacillus species have no synteny for the Fab operon. So they're completely missing this operon. And you'll note that Lactobacillus delbrueckii actually does have synteny, but I think there's been a lot of discussion in the field because it is quite divergent from other Lactobacillus species that it will probably receive a new taxonomy soon, and it's actually not something that is strictly associated with the urogenital microbiome. It is more of a generalist and is present in other environments as well. So this led us to the hypothesis again that urogenital lactobacillus species, because they're lacking these genes and they show the phenotypes and the growth assays that we saw, cannot perform de novo fatty acid biosynthesis. So we decided to test this hypothesis using metabolic tracing analysis. So we use C13 labeled glucose and a universally labeled glucose, and we fed it to two members of the Lactobacillus genus and members of the Lactobacillus and Lactoplantibacillus genera. And then we looked to see if they were basically able to incorporate the carbons from C13 glucose into the fatty acids of their phospholipids. And so we did this analysis, and we first cultivated Lactobacillus rhamnosus as a representative of the Lactobacilli, which does have the Fab operon. And we also cultivated Lactobacillus crispatus as the representative of our Lactobacillus, our urogenital Lactobacillus species, in CDM with C13 glucose and tween 80. And we have to include tween 80 because crispatus will not grow without tween 80 or without some source of fatty acids. And so we perform a lipid extraction and we do a fatty acid methyl ester analysis via GCMS. And so when we do this analysis, we see that for Lactobacillus rhamnosus, we see because tween 80 is present, there's about half of the fatty acids that we're able to isolate are labeled with C13, whereas none of the fatty acids that we isolate from Lactobacillus crispatus are labeled with C13. So this really strongly supports our hypothesis that Lactobacillus crispatus cannot perform this de novo fatty acid biosynthesis. And we can further break down actually the how much of the fatty acid is actually labeled. If we're looking at 18.1 here, how many carbons within that are actually have the label. So again, we find that the fatty acids in L. rhamnosus are the only ones that were labeled by C13. And we repeated this experiment with Lactobacillus gasseri, and we found the same results as Lactobacillus crispatus, where none of its fatty acids were labeled. And so just looking more into the data, because actually if you look more closely at the data, it always gets more interesting. And so in the presence of 280, like I said, Lactobacillus rhamnosus incorporates unlabeled oleic acid from 280. So this also kind of supports our hypothesis, where we can see this unlabeled oleic acid peak here that, is actually utilizing the oleic acid from 280. And then it's synthesizing, actually, really interestingly, oleic acid from labeled glucose. So, this labeled peak that we see here is actually oleic acid. So that's really interesting to us, especially. And with L. crispatus, you can see, we see when we look here at the trace, that there is, in this oleic acid peak here, we actually don't see any mass shift, and there's no C13 incorporated, and we actually see a little tiny oleic acid peak, and it's not labeled either. So there's basically no label at all in our L. crispatus fatty acid methyl esters. And so we further looked at rhamnosus, because rhamnosus can grow both with and without tween, obviously, because we don't think it is an auxotroph for fatty acids. And so when we grow in the presence of tween, we see, again, we saw that we see unlabeled oleic acid, which we think it's incorporating from the tween, and then all the label is actually in oleic acid. When we cultivate it without tween, kind of further supporting this idea that they're actually synthesizing oleic acid, is that when we grow without tween, the peak that we see primarily is this oleic acid with C13 incorporated. Okay, so we further wanted to know which exogenous fatty acids L. crispatus can use. So we know it can use oleic acid, but we wanted to know if it could use fatty acids of other chain lengths or saturations. And so we went ahead and did these experiments where we grew our Lactobacillus crispatus in CDM, supplemented with all of these different fatty acids from myristoleic acid, palmitoleic acid, oleic acid, and iconosaic acid as our unsaturated. And then we had some representatives of saturated fatty acids like palmitic acid and myristic acid. And we can see basically that that Lactobacillus crispatus seems to really prefer oleic acid, but it can grow on palmitoleic acid and maybe even on icosanoic acid. And this is another strain of Lactobacillus, and we see here a similar pattern, but different kinds of effect sizes, but it still seems to prefer oleic acid. And so we repeated these experiments with Lactobacillus gasseri and got very similar results, where we can see the maximum growth stimulation happening with our oleic acid. So it seems like both L. crispatus and L. gasseri prefer 18-1, but I think I'm convinced that at least some strains of these species can utilize 16-1 to some extent. And so our next question was, we were testing for the unsaturated fatty acids, we were testing cis fatty acids. And since we saw that in our tracing experiment, rhamnosus was actually making trans 18-1 or oleic acid, we wanted to know if L. crispatus could actually use trans fatty acids as well. And so we performed this experiment, same experiment as before with CDM, supplemented with either oleic acid, oleic acid, which is 18-1 trans at that 9th position, and then vicinic acid, which is an 18-1 trans at the 11th position. And we were able to see that it was able to utilize all three forms, both the cis form and the two trans forms that we tested, oleic acid and vicinic acid, for growth. And we were able, we repeated this, and we still have to do one more rep of LC 1700. We have to do another experiment with vicinic acid, but we are able to see that with oleic acid as well for a second strain. And then we, of course, did this for Lactobacillus gasseri, and we see the same trend where we are able to use both cis and trans fatty acids in the form of oleic acid and oleic acid for 2 strains of Lactobacillus gasseri. So we conclude that Lactobacillus gasseri and crispus can utilize 18-1 trans like oleic acid as fatty acid sources. And so then we next wanted to ask the question, if we feed them a trans fatty acid, is that what they'll incorporate in their membrane, or are they going to modify it to be a cis form, or is there going to be a mixture? And so what we did is we actually grew Lactobacillus crispus in CDM with either oleic acid or vicinic acid as a fatty acid source. And we isolated the lipids and quantified the fatty acids. And we found that Lactobacillus crispus grown in vicinic acid contain vicinic acid in their membranes. So their membranes, we have this blue peak, that's from the Lactobacillus crispus grown in vicinic acid. Their membranes are composed pretty much entirely of vicinic acid, where L. crispus grown in oleic acid contains oleic acid pretty exclusively within their membranes. And so I think this is really important because it basically reflects how the growth medium, right, when you have these types of auxotrophies and they're directly incorporating what's present for them in the growth medium into their membranes, might actually change, right, the biochemistry of the cell and the biochemistry of the membranes. And maybe we'll see different phenotypes, right, if we are providing them with different nutrients, and in this case, different types of fatty acids. And so I think thinking about what's in the media, obviously really matters, because these, at least in terms of fatty acids, these urogenital lactobacillus species really seem to be they are what they eat, And so we, the last kind of experiment that I'll tell you about is that we had done as part of a different study. We did quantitative metabolomics on human urine, and in doing that, we actually found that there were actually a ton of different lipid species like ceramides and glycerophospholipids present in human urine. And the reason we were kind of thinking about this, because we were like, well, where, in the context of the bladder are these lactobacillus species actually sourcing these fatty acids? Where are they coming from? And so it was encouraging to see that, of course, that these lipid species are present in urine. So next, we wanted to ask the question, if these urogenital lactobacillus species can actually use host lipids as a fatty acid source. And so as kind of a first experiment, we isolated lipids from 5637 bladder epithelial cells, and we basically just fed them to Lactobacillus crispus and gasseri in lieu of a pure oleic acid. And so when we did that, we actually got the nice result that that these 5637 lipids were able to stimulate growth of both Lactobacillus crispus (LC195) and Lactobacillus gasseri (LG637). And so we are still optimizing our extractions on this, and we're now performing GCMS and LCMS to identify the host fatty acids and actually the lipids that are incorporated in these membranes and that are present in these extracts, to really nail this down and see which what lipids exactly they're able to incorporate. So we're really excited to see the direction that this will go. And so I think the reason that this that thinking about these types of things and what the bacteria are actually going to be eating in their environment is really important as we think about using probiotic approaches to restore the microbiota in the treatment of different diseases. And so in our case, we think about recurrent UTI. And of course, one of the big things that we think about are, can we use probiotic approaches to actually restore a beneficial urinary microbiota in women with recurrent UTI. And so we think of this scenario where recurrent UTI and antibiotics disrupt the microbiota, and then maybe we can use probiotics to push it back to a healthy state. And so I think in the light of this research, my answer to this question actually is perhaps, but not if they don't have the correct nutrients available in the urogenital niche. So we also have to think about not just putting back the good bacteria, but we have to make sure we can modify the chemical environment so that they have what they need to proliferate and survive within that space. And this is, a very common thing, I think, throughout probiotic research in general and in different niches in the body. But it seems like it's also really important here, especially if our good bacteria that we want to put in seem to have very specific nutrient requirements. And we have to make sure that those are present in the niche that we're reintroducing them to. So I'd like to thank the stars behind this work. So this work has been really a work of passion by my postdoc, Dr. Michael Nugent, who is the best postdoc in the world, and a graduate student, Rishikesh Dalvi, who is nothing but amazing. They've really worked together to bring about the majority of all these data that you see. But we've also had tons of contributions from my graduate student, CJ Sands, who's on the bioinformatics side and is furthering our understanding of this in the evolutionary space as well, looking beyond even the genera that we've looked at to figure out how this was lost or gained, this activity of being able to make your own fatty acids. And then tons of undergrads and as well as high school students as well, since this is the only BSL-1 project really in my laboratory. And so then I'd like to thank the whole lab and my funding, and I'd be happy to take any question, and of course my collaborators. Thanks.